Hello, biology. Welcome to class. This is strange, isn't it? Here's a video explaining section 13.1. Please follow along in your books. I also encourage you to actually read the sections because we're not going to have as much in-class time to talk about specific things. So you are going to have to take some responsibility and take a bit of time of your own time to read. So just like in class, I'm going to go ahead and start talking through some of this stuff, and we can just go for it, I guess. So we're in chapter 13, talking about some genetic technology. We've learned about genes, we've learned about heredity and stuff like that. Genes being passed down. We got Punnett squares and complete dominance and incomplete dominance and homozygous recessive things and all these words that hopefully you all remember, because if you don't, this is going to be kind of a tough chapter. Although, maybe you guys will be happy to know if you look at how much vocabulary words are in this section. There's only 2. We've already talked about this first one quite a bit. The second one, I guess, is kind of new, but it's going to be really easy. So 13.1 is just talking about applied genetics. These first couple paragraphs are just reminding us that as humans, we have kind of been messing with animals for as long as possible. And what I mean by that is we have 2 animals, we breed them together to make a stronger one, right? So we have a family of cows, and we breed the strong cows to have more strong cow babies to pull carts and make milk and stuff like that. Humans have been doing that for quite a bit of time. Same thing with plants. And even before we even knew anything about genetics, we've been doing this stuff. Here we see, since ancient times have been doing that. The juiciest berries, the most disease-resistant clover, best milk-producing cows, the best egg-laying hens. Like we want the best types of animals for this stuff because they're the most useful for us, right? This is actually kind of, I don't want to say this specifically is genetic engineering, but it's kind of like the base level for genetic engineering. We are kind of, it's not natural, right? We're selectively breeding these people. That's what this means. We're selectively breeding these organisms for their offspring to have specific traits that we want them to have. That way they are the most productive for us. In fancier words, we are increasing the frequency of a desired allele within a population, right? If there's something desirable in an organism and we breed that over and over again, then we have increased the frequency of that specific trait, of that specific allele, within a population of whatever we're talking about. Does that make sense? The book says you can read about the selective breeding of domestic cats on page 1066, which I didn't even know there was a page 1066, so I looked in the book. I don't know if you can see, but look at that cat getting its makeup done. I thought that was pretty interesting. I laughed a little harder the first time I saw it, but that was like 10 minutes ago during the first time I tried to record this and I messed up, so it's not as funny to me anymore. Specific example we got right here. In 1947, an average cow produced about 5,000 pounds of milk. In 1997, the average cow produced almost 17,000 pounds of milk. That's more than 3 times the amount of milk per cow, right? 5,000, 17,000. That's more than 3 times the amount of milk. That all comes down to pretty much just selectively breeding. We just keep breeding all the families of cows that make a lot of milk until we have these crazy cows that can make 17,000 pounds of milk in 1 year. That's crazy. That means you need 3 times less cows, right? Because 3 of these 1947 cows can't match up with 1 of these 1997 cows. And that was, what, 23 years ago, so you can assume this is a little better at least, if not a lot better. That's crazy. And that's just breeding. That's just picking the best ones to get the best traits out of it. Dogs, let's see. I have one of these dogs, and I'll probably bring her in for a video because why not? And these dogs are bred because people liked certain traits, and they kept breeding them until they got this, and now they look like a specific thing. Another dog. Hey Jacob, what's an example of a dog? Correct, Jacob. Thank you for your help and volunteering and talking to your computer screen by yourself. A German Shepherd. Cool. Another example, I mean, the Boxer and a German Shepherd are a dog, right? But we have selectively bred them to bring up specific specific traits that are useful. Carlos, what about you? You got a dog? Ooh, weird. That's a I didn't know you'd know that dog, but look at how gigantic this dog is. Good job, Carlos, on picking one of these dogs. Look how terrifying this thing is. Look at that. Look at how gigantic this dog is. But we have bred dogs to be like this. We have bred dogs to be like that by just selectively breeding the ones that have all these desirable traits. And that's called inbreeding. So we would have had to, back in the day, start breeding big dogs and keep breeding big dogs until we get something that looks like this. And we keep breeding the same lines over and over. That's what's called inbreeding. We see that Boxers, look at how old is this? 1920s to 1930s, they almost look the same, right? We've been breeding these dogs to look the same for 100 years now. We keep inbreeding them. What that does is it results in offspring that are homozygous for most traits. Because they keep breeding, they keep getting the same kind of traits showing up. And that can be good and bad, because we also talked about inbreeding can bring out harmful recessive traits. Because now, if you breed a German Shepherd with a German Shepherd, they both are probably carrying bad recessive genes in them, and now they are probably going to have offspring with that recessive gene in there. Does that make sense? We do this with horses, we do this with dogs, we do this with plants. Although in plants it's called a cultivar instead of a breed, but it's the same thing. We breed them for specific characteristics, and that means they're homozygous for lots of different things. Homozygous for lots of different traits, including harmful recessive traits. And remember, if you have two of the lowercase t's for some type of recessive thing, that means bad things happen. That means the recessive traits are shown. Still working with that. Okay. So plants are a little strange. We talked about how plants don't really care about non-disjunction, which means plants don't really care if sometimes they get a little more chromosomes than they're supposed to. They actually kind of get stronger. Well, it turns out plants are also really good at, when we crossbreed them, they get really good at becoming larger, producing more food, becoming more disease resistant. And when two cultivars or two breeds are crossed, their offspring will be what we call hybrids. Hybrids produced by crossing two purebred plants are often larger and stronger than their counterparts. Many plants that we need today, it says here, wheat, corn, rice, flowers, roses, they've all been developed by hybridization, meaning we take two strains or two like families, cross them together to kind of combine all the best traits of both, and interbreed that until we get a super crop, a super corn plant or a super wheat plant. And this is not even counting the whole non-disjunction stuff that we were talking about. This is just literally just having certain wheat plants next to each other and make more wheat plants that are stronger. So all this is good and everything, but the question is, how can we even tell what the organisms are, right? Like, you can't look at someone and tell what their genotype is. You can tell what their phenotype is, what they look like, but you can't tell their genotype in all cases. So if you want to make sure that when you breed two things that they have the correct genes you want, what you actually have to end up doing for small animals or for plants is to do a test cross. So one way to determine the genotype of an organism is to perform a test cross. It's a cross of an individual of unknown genotype with an individual of known genotype. Okay, so you pick one organism that you know what the genotype is. Typically, it's homozygous recessive, because remember, that's really rare. So if you have an organism with that, if you have an organism with the if you have an organism with 2 lowercase letters, you know that that's rare. So the organism has these 2 T's you're not unsure of. But remember, if something is showing a dominant trait, we don't know if they're like this or like this, right? Because this will look the same to us. So we're not sure what's happening over here. So we have to do a test cross. Usually it's with one homozygous recessive and then with one of these 2. But we don't know. That's the point of it being a test. We are crossing this with one of these to try to find out what we can get out of this. Many traits, such as disease vulnerability in roses and progressive blindness in shepherd dogs, are inherited as recessive alleles. Remember, you keep breeding the same plants, they're going to keep getting the same traits. You keep breeding the same dogs, they're going to keep getting the same traits. Some of your dogs might have joint pain, and that's probably a recessive trait that they have inherited because of their breed. And that's also why, in my opinion, dogs that you don't know what kind of breed they are seemingly last a little longer than purebred dogs, because you kind of smooth over all of the recessive genes. I don't know about you, but most of the families I know, if they have a mutt, that dog lives until it's like 20, and it doesn't ever have any health problems. It just gets old and dies, compared to other dogs, like German Shepherd dogs might get blind, boxers get joint problems and stuff like that. So, all right. So what are the possible results of a test cross? If a known parent is homozygous recessive and an unknown parent is homozygous dominant, we wouldn't know that, though. That's what the unknown mean. All of the offspring will be heterozygous. So that's what's happening over here. If we know that this parent right here is homozygous recessive and the unknown ones happens to be homozygous dominant, then to the Punnett square, we all get capital D, lower d, capital lower, capital lower, capital lower, which means all of the offspring, if these are originals, all of our offspring will be capital D, lower d, so they'll look tall. On the other hand, if we do a test cross with our known homozygous recessive with a heterozygous, we wouldn't know that, though. We're not sure what this dog has. We're just crossing it. What happens is there's a 50-50 chance. Half of their kids will be lowercase and half, because it's short, half of the dogs will be 2 lowercase d's and half of the dogs will be uppercase lowercase d. Right? So half of the dogs will have the phenotype of the dominant trait, and half of the dogs will have the phenotype of the being short. The problem with test crosses is that you can't really be sure what you have unless you have a lot of offspring, because it's possible for you to cross this with this and only get tall dogs, right? Because it's a 50-50 chance. If you only have 2 dogs, the 50-50 chance of getting 2 tall dogs is pretty, that's doable. So it only shows up once you have a lot of offspring, and then you can really tell, then you can really try to deduce what the phenotype and the genotype were of the parent, of the unknown parent. So that might be why it's unfeasible for humans to do test crosses. If you want to know if you're going to pass down a certain trait, the only way to tell is to have a bunch of kids, and that's not really ethical to have a bunch of kids to see what kind of kids you can have. That doesn't make any sense. We can do it with dogs and hamsters and stuff like that because their life cycle is pretty short. So we can do that. We can do that with plants because, who cares? You can make a bunch of plants and they're not good, just kill them or sell them, whatever, and just make more. But we can't really do that with humans. And that actually comes up in your homework over here. So if you made it all the way to the end of this video, you will probably know the answer to number 5. Because it's unethical and it takes way too long for humans to be able to even go through this process. And that's about it for 13.1.